Thank you for that introduction. Dr. Gilfix has put me in a very awkward position. I now stand between you and lunch, and he overran, so I'm expecting... Okay, I was expecting an angry mob. Okay, so actually the title that was listed in the literature has two inaccuracies. One, it says there's only one clinical trial. I'm about to show you something that's also in a clinical trial. And that was the title of the grant that our last grant from HCU America. What I was actually going to talk about is Blood, Brain and Bone, New Insights into Homocystinuria Pathogenesis. As Ferdinand was saying earlier, we want to know about pathogenic mechanisms so we can design treatments. But we're going to go one step forward and use that pathogenesis to illustrate a treatment for you. So, how many people here are patients or family members of patients? Okay, so that is going to dictate how I'm going to do this, because I wasn't sure if this was going to be, but once we reconstituted the Berlin Wall over there, and I honestly think the last talk made no allowances for the general public whatsoever. I'm going to try and keep this as simple as I can. And that is part of the reason I think that's actually probably true. If you can't keep it reasonably simple, you probably don't have the best understanding. Okay, so a slightly more layman term for the title is how things go wrong in homocystinuria, how to improve treatment, and why individuals with the same mutations may not get, or may get different outcomes. Okay, I'm not sure what that was. I guess it's not essential because I'm still talking. All right, so what, so I can drop it again? What, scientists, we believe in replication. I'm just going to keep smashing it till you take it. Okay. So conflict of interest, declare funding. Will is over there from Taurine, and they are running the clinical trial. I also work with Petra Bio. They can't be here for this time, but that project with methionine degrading bacteria is still active and NIH funded. And we've had some minimal supply for funding from the NIH, but some from the state of Colorado, and HCU America and the William Hummel HCU Research Fund, for which I'm very grateful. Okay, so I sometimes show this to what we actually do and the methods that we approach. And I was once offered a job in England to return, and they sort of said, could you give us a slide that just shows everything that you do? And so that's what I did. And now you may have noticed all of those diseases on the left. Anyone who's a parent here knows you don't play favorites with your children, but that's not the case for diseases. And the vast majority of my time is spent on HCU and remethylation disorders. Okay, so I think most of us has been covered for most of you. Obviously, classical homocystinuria is caused by a mutation in cystathionine beta synthase, and that causes, actually, whenever I put this up, I'm always reminded, I've got a colleague at Houston, Dick Fennell, who says, you might as well put it up in Chinese, for they're so complicated. So in case you're interested, okay. But you all know that CBS is the mutation that gets mutated in classical homocystinuria. That leads to severe elevations in homocysteine, methionine, severe elevations in Adamet and S-adenosyl homocysteine, which has some implications for the work you described earlier, because Adamet actually inhibits methylene tetrahydrofolate reductase and stimulates CBS. So you may well, anyway, we can talk about that later, but those are underestimated. I actually believe a lot of the epigenetic effects in homocystinuria are because of the S-adenosyl homocysteine, which inhibits a wide range of cellular methylases. You also get downstream effects. So you've got a decrease, in theory, in endogenous ability to make cysteine, and a decrease in cystathionine. Okay, but it is important to realize that we are not just walking buckets of plasma, and that most of what we know about metabolic changes in classical homocystinuria come from blood samples, because that's what you get from patients. Pinning them down and taking lumps of tissue out of them is not viable. So it may well be that if we just look at blood, how we look will dictate what we find. And it may give us an inaccurate picture of what's going wrong, and that's why we need animal models. And much of what's going on in classical homocystinuria is happening deep in tissues where the gene expression profiles, the genes that are expressed, and the biochemical changes could be very different from what we see in the blood. And we've found that classical homocystinuria causes a much wider range of metabolic disturbances than those shown in the previous slide. And those disturbances will vary wildly depending on the specific tissue being investigated. So this should be familiar to all of you. Okay, these are the clinical abnormalities. The eye and the skeletal abnormalities are usually lumped together as Marfanoid habitus because they look a lot like Marfan syndrome. And I'm going to come back to that later. Before CBS deficiency was discovered, it was often misdiagnosed as Marfan syndrome. In terms of mortality, the problem is really vascular. You get thrombosis. There's a 30% chance of having a stroke before 20 if you're not treated. And obviously, in terms of quality of life, cognitive impairment, if you don't get early treatment, you can be quite severe. So, current treatment. If you're lucky, you've got a pyridoxine responsive form of the disease, although partial response is also possible. I've been talking to some people about that recently. For those that are not partially responsive, it's twofold. One is you get rid of the precursor of homocysteine, which is methionine in the diet. So you have a low protein diet. And you also use betaine, which is also trimethylglycine. And what that does is it sticks a methyl group onto the homocysteine to convert it back to methionine away from homocysteine. So that's how it lowers the homocysteine. Okay, and I'm going to use this. That would be the BHMT there. And so that would add a methyl group to the homocysteine to make the methionine, making another chemical called dimethylglycine in the process. Okay, I think most people will agree that the methionine restricted diet is the worst part of this. It's actually very, very hard, especially if you're diagnosed late and you know what normal food tastes like. It can be very, very hard to adapt to this. And so this is something I've been trying to do something about. And yeah, I don't know if any of you have heard me talk before. I usually have to explain this. We work with a mouse model that we generated called the human-only mouse model. Obviously, you can tell by my charming accent that I don't come from here, so I didn't know what ho meant when I actually named the mouse. And the joke I always make is, well, if I must be the best pimp ever because all my hoes have fur coats. So what it has is it has the human gene, the human CBS gene, and no mouse CBS gene. And by a lucky accident, that human gene is not expressed very highly. The classical CBS knockout mouse has terrible liver problems, far more severe than humans. And the mice don't live very long. And the liver is so badly damaged, it affects coagulation. So it's a terrible model in many ways, although we did find a use for it, which I'm going to talk about. But by pure luck, because I've got pointed ears, by pure luck, low expression of the human gene rescued the liver phenotype, but kept everything else in place. The biochemistry was actually really good. Now, it's true that not all mice models are an accurate recapitulation. They don't always make a good model. But the hoe mouse is actually very good. It's got a hypercoagulative phenotype, so it means it clots very, very quickly. It responds biochemically, and it also improves its outcomes with betaine treatment. It's got impaired learning and memory. It's got decreased bone density. I'm coming back to that. It's got inflammation. It's got all kinds of things. But what's really interesting is it's telling us stuff that we didn't really know. So how do things go wrong? We're investigating a lot of different candidate mechanisms. I don't have time to go through all of them today, but I'm going to go through some of them. But basically, if nothing else, you should be aware of the fact that we are working diligently on multiple mechanisms and working very hard to develop new treatments. But as we begin to understand how things go wrong in homocystinuria, we then start to think, how can we leverage that knowledge to design better treatments? And I'm going to give you two really interesting examples. So we are currently developing 5 different therapeutic strategies for improving treatment. And one of the fundamental things in this is oxidative stress as a central pathogenic mechanism. And okay, I thought I had another slide before that, but okay. But one of the things that we were interested in was, does oxidative stress promote thrombosis in classical homocystinuria? And when you want to actually, the simplest way to assess thrombosis in a mouse is to clip off a certain part of its tail, a certain measurement, and then you put the tail into warm saline and you time how long it takes to stop bleeding. So that's its coagulation time. If you look here, this is basically, it's a graphical. This is how much blood a normal mouse will dump into the tube of saline before it clots. This is what it looks like with the homocystinuric mice. They clot about 3 times faster, very, very quickly. And that is what we call a hypercoagulative phenotype. Now, there are 2 phrases that I think are probably the most misused in the English language. One is research has shown, because most of the time it's just people making up their own opinions and saying, well, this research has shown. But antioxidants is in that category. A lot of people say antioxidant, and if I didn't have a conscience, I could probably be a multimillionaire talking about antioxidants on late night television and selling you something that doesn't work. But as he says, I don't know what oxidants are, but I'm against them. Okay, so taurine is basically, as well as being blocked in their ability to make cysteine, homocystinuric patients can't make their own taurine either. Cats can't make it either, which is why if you put a cat on a vegetarian diet, they go blind fairly quickly. But it has many cellular functions, and it can actually act as an effective antioxidant by sparing more cysteine for glutathione synthesis. Glutathione is the major cellular antioxidant. And it sticks around much better than cysteine does. The problem for the patient population is you're blocked in your ability to make taurine for yourselves. And most of the foods, like meat and seafood, that are full of taurine are also full of methionine too. So there's a good chance you're not getting enough taurine. Okay, so we had, in a series of papers, found that taurine metabolism is disrupted in homocystinuria. We found that it was a powerful antioxidant by sparing the glutathione, as I mentioned, and that it improved the efficacy of betaine treatment. And then we had a human clinical trial, and we found that it worked very well in terms of having a beneficial effect on the way the blood vessels respond to the stimuli that open and close them. It's called endothelial dysfunction. And what we found was also that, which one is it? Yeah, that taurine was actually very effective at getting rid of the hypercoagulative phenotype. So what we have here is the blood from normal mice who have had their tail clipped. This is the very clear blood from the homocystinuric mice with no treatment. So they're clotting very quickly. And if we gave them taurine, basically they look a lot more like the wild types. And their coagulation times are very similar to the control mice. So it was actually quite effective. Endothelial dysfunction is primarily, there's a lot of different stimuli that will actually cause endothelial dysfunction, like diabetes, hypertension, smoking. Pretty much everything your doctor will tell you is going to cause you cardiovascular problems. And if you get it, you can end up with an inflamed state, impaired vasomotion, increased thrombosis. Okay. But what we found in the clinical trial was those patients, the classical homocystinuric patients that showed endothelial dysfunction, had it completely normalized by the taurine treatment. And critically, it didn't affect endothelial function in people who didn't have the problem. So it's almost like a dream drug. It's a highly safe compound, and it fixed the problem, but only when it's actually there. Okay. And we've now done in research that funded by HCU America, we've now shown that it does that, we think, by affecting the expression of an enzyme called CYP4A that inhibits the production of a compound called nitric oxide. And nitric oxide is a gas that provides second-by-second regulation of your blood vessels and their tone, and your blood pressure, and they open and relax depending on how much nitric oxide is there. We found that the mice, homocystinuric mice, have very low levels of nitric oxide, but that the taurine completely normalized it. So we think that that's actually quite useful. Okay. But the question is it all just oxidative stress? And the answer to that is no, it's not. And that's where we come to one carbon folate metabolism. Although there is a link between the two because of the role of one carbon metabolism in oxidative stress, but we'll just keep it for one carbon metabolism for now. So we've already done that. Okay. So again, I'm motivated to try and reduce the dependence upon the methionine restricted diet, because that cat's expression is probably what it looks like if you have to eat one. That's what the cat looks like if you give them a salad. Okay, so when I talked to Harvey Levy about this and about betaine and everything, I said what I was working on. He said, yeah, that's great, because betaine is fantastic initially, but it doesn't last. Its efficacy wears off over fairly, within about 6 weeks or so. So we thought if the efficacy of betaine treatment could be improved, we thought methionine restriction could be relaxed, leading to a significant improvement in patient outcome and quality of life. I then ran into a lot of negative stuff in a review from the late Dr. Harvey Mudd. I know it's Dr. Harvey Mudd, even though reviewers are supposed to be anonymous. But he would sort of say, when anyone knew that they'd been reviewed by him, because he would say, well, you haven't considered Hesketh in 1927. I thought there's only one man alive who's read that paper. And he's not alive anymore, unfortunately. But he basically said, well, depending on what we know about folate metabolism, betaine can only ever be an adjunctive therapy. And my take was, that's all been proven in normals, in wild types, in normal biochemistry. If you take a gene out of the picture, all bets are off. You don't know what's going on in one carbon metabolism and homocystinuria. And then I realized I don't either. So I thought I need to do something about it. Okay, so a few reasons made me think that there was that betaine could be improved. When we have treated our mice with betaine, the homocysteine goes down really nicely for the first week. And then after 6 weeks, it's back up to a dangerous level. It's not back completely, but back up, which means something has changed between those two time points. And if we understand what's changed between those time points, maybe we can get it back to the original level. Betaine treatment works very poorly in patients with MTR deficiency here, okay, because they have what we call a methylfolate trap. This form of folate is trapped because it can't be converted into this one, okay? But this form of folate, tetrahydrofolate, is required for these reactions here, downstream from the betaine reaction. Oops, go back to that. And yeah, it's that. Okay, so something you need to know about betaine homocysteine methyltransferase. That's the enzyme that allows betaine to work. It takes the betaine, it takes a methyl group off of there, and then it puts it onto homocysteine to make methionine, but it also produces this compound, dimethylglycine. And basically, but that compound, dimethylglycine, sticks to be the enzyme, betaine homocysteine methyltransferase, and inhibits its activity. So if you don't clear this compound, this isn't going to work very well for very long. So the removal of dimethylglycine is the choke point, okay? If you think of this as the engine converting homocysteine to methionine. This is the exhaust. If you block the exhaust pipe on your car, it's not going to work very well for very long. Okay, so, and as I said, methylfolate trap. This is what CBS deficient mice look like on betaine, and at 1 week. So it's very good. It's down to about 50 micromolar. At 6 weeks, it's back up to 100, or over 100, sorry. And that's when the problems come back in terms of blood clotting and other problems. But this is a MTFR deficient mouse, and it has about 70% reduction in methionine synthase. And as you see, the reduction in homocysteine is about 15%, which is what we see in human patients. So it's pretty rubbish. It's not actually that useful. Okay, so despite the crucial role of tetrahydrofolate in the remethylation of homocysteine, oh, actually, I think I should have gone there. Okay, yeah. So this enzyme here dimethylglycine dehydrogenase is the one that's responsible for removing the exhaust from the engine. If you can get rid of all of this, this will go at top speed. But this, whoops, excuse me, happened. But for that reaction to happen, it requires this species of folate. It won't work very well without this. Okay. In fact, it may even make formaldehyde, but that's a separate story. Okay, so, and there's been like one previous report of subnormal serine folate levels, increased folate clearance, and a requirement for folate therapy in humans, subjects with HCU back in 1968 when I was about 4 years old. And since then, virtually nothing in this particular area of CBS deficiency. It's complicated, okay? It does a lot of these things. It does methyl groups, which Ferdinand talked about earlier. It's involved in DNA synthesis, both purines and thymidylate synthesis, metabolism of multiple amino acids, and there's a fifth function for oxidative stress as well. Okay, so this is the more complicated version of the folate cycle, and it talks about this. Within the cell, there are different environments. There's the cytoplasm, there's the nucleus, and there's the engine, which is called the mitochondria, which is where a lot of energy metabolism happens. And tetrahydrofolate cycles through multiple forms of different folate species. Oh, damn, sorry. And then also, one of the things that you see here is formate as basically a carrier of folate molecules between these subcellular compartments. And sorry, this thing's on a hair trigger. But it's really quite interesting that basically serine is an important part of this. And this part is designed to break down serine to make formate, and this part is designed to rebuild it, but also to provide this material for DNA synthesis and cell growth. But it is very complicated, and that is why people have kept the hell away from it. Okay, I'm cynical, but I think people have thought, yeah, we should have a look at folate metabolism and homocystinuria. They take one look at that and think, yeah, maybe I'll just move on to something else, because it is difficult. So, but if we start with a hypothesis, if folate metabolism is disrupted and depleted in homocystinuria, what should we see if we look at the biochemistry of the liver, for instance? Well, one of the things you should see is disrupted purine metabolism. That means disrupted DNA synthesis. And this is what, if you starve mice for 6 months of folate, they get this, basically widespread problems with their purine metabolism. Lovely. And we get almost identical patterns of the same chemicals in the HCU mice, okay? So that's an indirect sign that there is a problem with the folate supply. And these are two enzymes that are involved in purine biosynthesis, and they are severely altered in homocystinuria. And we have now done every single enzyme that you see on there that's in the black. We have now analyzed its expression in control mice, homocystinuric mice, and in the presence and absence of treatment. And it is exhausting. But one enzyme I want to draw your attention to, because I think it is the daddy of a lot of this, is dihydrofolate reductase. Okay, that's this enzyme here. Dietary folates that come from your food are not much use to you, but this enzyme will convert them into dihydrofolate and tetrahydrofolate, and the whole circus can start from there. This enzyme was the very first chemotherapy target. If you block dihydrofolate reductase, it makes it very hard for leukemia cells to make DNA, and therefore easier to kill the tumor. Oh, sorry, not tumor, the cancer. Okay, so one of the problems with that for classical homocystinuria is we found that the dihydrofolate reductase, that critical enzyme for making these folates, is strongly repressed, both at the protein level and the enzyme activity level. Okay, and critically, when we treat it with betaine, which is when we really need the folates, the repression was not lifted. I think you can agree it's not subtle. This is the level of protein that we see in normal mice for the dihydrofolate reductase, and this is what it looks like in the homocysteinuric mice. Okay. Okay, I'm going to talk about that in a second. So, we then were interested, I'm going to just talk about some other enzymes that make tetrahydrofolate, or critical determinants. And there's one in the cytoplasm, and it's got a different version in the mitochondria. But as you can see, it's strongly repressed. That is ALDH1L1 and 1L2. Okay. But we got the first hints that this may also be related to oxidative stress, because when we treat with betaine, it comes back a bit. The mitochondrial one doesn't. But if we treat with taurine, oh bloody hell. The one on the graph C and F, that's that enzyme that makes 10-formyl tetrahydrofolate and generates tetrahydrofolate. It is strongly induced by taurine. Okay, and there is no real explanation for why there would be a crossover effect between 1-carbon metabolism and sulfonic acid metabolism, but it's most likely going to be an oxidative stress thing. Okay, so that's another one that's probably limiting the amount of tetrahydrofolate that is crucial for the betaine reaction. So we asked, well, what else can there be? And something that's becoming an emerging field is that the molecules that we have in our bodies are actually sensitive to oxidative damage. They can be broken down, and that they can also be repaired, and that it's critical for some molecules that are very fragile, very labile, to be repaired constantly. And so there are 3 currently known enzymes that can repair folates damaged by reactive oxygen species. One of them is dihydrofolate reductase, and that has 2 known repair functions. And we've just shown that it's strongly repressed. So you've got decreased capacity for repairing folates. MTHFS and QDPR. Okay, MTHFS will actually repair oxidized 5,10-methylene tetrahydrofolate. And that's important. I just want to draw your attention to this thing here. 5-formyl THF is folinic acid, or leucovorin, and it's sometimes given as a supplement to homocysteinurics. But as you can see, the enzyme that actually converts it into something, there's no use to 5-formyl tetrahydrofolate. It doesn't do anything. But it needs to be acted on by this enzyme to go into the normal folate metabolism. And the problem for that is it's not actually... it's repressed in homocysteinuria, and that repression is not lifted by betaine. There's one other enzyme that acts basically on tetrahydrofolate itself. That gets oxidized to a quinone derivative, but QDPR can rescue it. It can repair it. But QDPR is also strongly repressed in homocysteinuria, but that does come back a bit with betaine. Okay, so what about catabolism? All the way through your life, molecules in your body are being broken down. And there are processes for breaking down folates as well. And the critical enzyme in that process is an enzyme called NAT2. And unfortunately, that's induced. So as well as your decrease in making tetrahydrofolate, your decrease in your ability to repair it, you've also got an increase in breaking it down. So by now, it's starting to look like a conspiracy. You've got decreased tetrahydrofolate production, strong repression of every known mechanism of folate repair, and increased folate catabolism. So it looks like a conspiracy to deplete folates in general and tetrahydrofolate in particular. And given the relevance of tetrahydrofolate to betaine treatment efficacy, we had a look at what the folate levels look like. And dihydrofolate is way down in the liver. Tetrahydrofolate is way down in the liver, and formate is down in the plasma. So with betaine treatment, we may well have been driving with the brakes on for the last 50 years or so. So I'm going to run out of time. I'm going to leave this bit. Okay, so if the lack of tetrahydrofolate is the problem for betaine treatment in HCU, how can we fix it? Okay, so We did try folic acid, which was mentioned earlier, but folic acid is actually a synthetic folate, and it's only useful if dihydrofolate reductase, that enzyme, converts it into DHF and THF. So it didn't work very well. And we tried the leucovorin, and that didn't work well, although at the time we didn't know why, but now we do. But then we thought about formic acid, and it's the simplest form of one carbon donors. And it will serve as a gradual release donor of one carbon groups to the intracellular tetrahydrofolate pool. So when we treated with the formate, we found that just formate alone significantly lowered the homocysteine in the homocysteinuric mice. When we combined it with the betaine, it had a dramatic effect, because this, ladies and gentlemen, is in the presence of a full methionine diet. That's 20 micromolar homocysteine. So that is quite a phenomenal result. 3 lines of evidence made us think it was turbocharging how the BHMT works, okay? First of all, we found that the choke point enzyme, the dimethylglycine dehydrogenase, is induced by the formate. So it promotes the clearance of the exhaust from the engine, if you will. And it's even more induced when it's with formate plus betaine. So much, so good. So basically the exhaust pipe has been unblocked, and so the engine can run at maximum speed. In CBS null mice, that's the classic knockout mouse where they've got terrible liver problems, the formate doesn't work because the BHMT doesn't work. The enzyme, the liver is so damaged, the BHMT doesn't work. They don't respond to betaine, and the formate didn't lower the homocysteine, okay? So if there's no functional BHMT, formate cannot lower homocysteine. Finally, some gentlemen in the Czech Republic collaborated with us and made this inhibitor. And this inhibitor basically inhibits BHMT. And if we co-treat BHMT with the formate doesn't work. So if we cripple the enzyme, the formate doesn't work. So that makes us think basically, we think that formate is turbocharging the BHMT response. How else might one carbon metabolism contribute to pathogenesis in homocystinuria? One of the things is there's an amino acid called serine. And this is a great example of the fact that we're not just walking buckets of blood. Serine is critical in the brain for development. It's an essential precursor for sphingolipids and ceramides. These are all brain essential compounds. And it's also converted into another form called D-serine, which is basically neuroactive. So it is a crucial metabolite. And we found with our metabolomics first that there's like a 70% decrease of serine in the brain of the homocysteinuric mice, but not in the liver, not in the kidney, not in the plasma. It seems to be brain specific. And when we looked at the sphingolipids, we found what we expected. We found sphingonine and all these compounds that are critical for brain development are all down. So this may well be a mechanism for cognitive impairment in classical homocystinuria. And so we did this by a technique called metabolomics, which is semi-quantitative. So we thought we should really measure this directly. So we measured L-serine, D-serine, and glycine in the hippocampi, which is the part of the brain that's for learning and memory. It's called hippocampus because of hippocampe in French is seahorse, and it looks like the same shape as a seahorse. It's probably from Latin rather than French, but you know, they claim everything. So L-serine was, I mean, it almost looks fraudulent because it matched the metabolomics so perfectly. It's like a 70% reduction in L-serine, a 70% reduction in D-serine, and a significant reduction in glycine. So yeah, we were definitely onto something. Then we thought, well, okay, we've got. I don't have time to show it to you, but we know the mice have a cognitive impairment. Does betaine correct this depletion of L-serine, D-serine, and glycine in the hippocampus? And the simple answer is, as you can see from this, no, it doesn't. Okay, it doesn't really come up, doesn't come up. Does formate help? And then the simple answer is yes. It pretty much normalized the serine level, L-serine level, D-serine level, glycine level in the brain of the homocysteinuric mice. So it's not just lowering the homocysteine, it's also correcting these other metabolic defects that we think are critical for cognition. Again, this is in the presence of a full methionine diet, okay? All right, so what about connective tissue disturbances in classical homocystinuria? Is oxidative stress or one carbon metabolism the problem? It could be both, because one thing I'd like to have everybody come away from here is that I think that our view of connective tissue disorders in classical homocystinuria is overly simplistic. Okay, everybody focuses on the marfanoid stuff, but the decreased bone density, and actually, you know what, I'm going to show you, I think I've got enough time. I'm going to show you a mechanism for the marfanoid thing. Okay, Marfan syndrome is caused by mutations in the fibrillin-1 gene. Fibrillin-1 is a large protein, and it's got a lot of cysteine in it. It's about 18% cysteine, and it needs that to form bridges that make it mechanically strong and hold your eye lens in place or keep your bones developing the way they should do. So consequently, homocystinuria is a fibrillinopathy as well as a metabolic disease. But you don't really get the Marfan syndrome type stuff in the other forms of homocystinuria, the remethylation defects. Okay, so we started off with the idea of, well, how could oxidative stress cause this problem? And we had this idea that there's greater oxidative stress in classical homocystinuria than there is in the remethylation defects. And maybe that's why they get the marfanoid problems that the remethylation defects don't. And if you get oxidative stress, you get lipids being attacked by reactive oxygen, and they form these compounds, lots of these kind of what we call aldehydes. They're interesting, but they will actually stick to proteins. They will actually stick to cysteine residues, and they will interfere with protein functions, okay? So we thought, well, maybe reactive aldehydes formed by oxidative stress induced lipid peroxidation have the potential to impair protein function in homocystinuria by forming covalent adducts, which is a posh way of saying they stick to cysteine, histidine, or lysine, those amino acids that are for cross-linking. And we had evidence in mice first. This brown stain here, those are aldehydes sticking to proteins. And this is a human homocystinuric patient liver. And the mouse predicted this. This was never known in humans, but we can see that lots and lots of the proteins in the liver are having these aldehydes sticking to them. It's not subtle in the kidney either. It's going on there, and it's going on as the cerebellum. So it is happening throughout the body. And we thought, well, how could oxidative stress induce marfanoid connective tissue disturbances? And it's fibrillin. And what we thought was, chronic oxidative stress would make these aldehydes stick to the fibrillin residues, to the cysteine residues, and thus mimic what would cause Marfan-like syndrome. These are fibroblasts, and what that is basically, these are control fibroblasts from normal humans, and these are from MTHFR deficient humans, and these are from CBS deficient humans. And this is one of the species of aldehydes we're looking for using antibodies to try and detect if these things are sticking to fibrillin. And what we find is there's no sticking in the control. There is no sticking in the MTHFR, but that red stain there is 4-HNE, 4-hydroxynonenal, that sticky aldehyde attacking the fibrillin that the fibroblasts are excreting into the media. We did mass spec analysis to identify which part of the fibrillin is getting these residues sticking to it, which is what that is. And then we identified the residues that it was sticking to. And that is, you see these cysteine residues? These are where the aldehydes are sticking. Every single one of these cysteine residues is known to cause Marfan syndrome if it's mutated. So that's how oxidative stress is causing HCU to mimic Marfan syndrome. There isn't a single residue that we've identified stuck in fibrillin with those chemicals that doesn't cause Marfan syndrome. Okay, so It's a plausible role for oxidative stress-induced aldehyde reduction as a pathogenic mechanism for marfanoid connective tissue disturbances. Okay. However, homocystinuric patients are actually different from Marfan syndrome in many ways in their bones. There's more to the connective tissue problems in homocystinuria than the marfanoid habitus. This is a paper published by my countrymen in 1972 that doesn't get enough, I think, doesn't get enough attention. And one of the things that they pointed out is that Marfan people don't get big knees, whereas it's quite common amongst some homocystinurics to have much bigger knees than normal. This is a knee, excuse me, that is a knee on the left is from a normal mouse. The knee on the right is typical and representative of our homocystinuric mice, and the knees are about 4 times the size of the normal control. But that's not found in Marfan syndrome. And then we thought, well, osteopathy, basically changes in the bone architecture and bone weakness and decreased bone density are far more severe in homocystinuria compared to Marfan syndrome, which we think indicates a different mechanism for the disease. And so the risk of bone breakage persists in homocystinuric patients, even in patients with good homocystinuria control. Some of you in this room may well be still breaking bones as adults in things that you know surprise you. So it's a significant problem. We think it's neglected, and so we decided we would have a look at it. Okay, so this is a little bit of basic biology for you. This is what one of your long bones looks like. This area here we call the trabecular region, and this hard bone on the outside we call the cortical region. Okay, we've now done a detailed micro CT computer tomography analysis of bone strength and architecture in the cortical and the trabecular regions of femurs, and that means the thigh bones, from 2 different mouse models of HCU, the hoe mice and the previously we thought useless CBS null mice. Okay, in both models, in the trabecular analysis, we noticed that 13 out of the 18 parameters of bone architecture were negatively affected in both models of homocystinuria. In the cortical analysis, 21 of the 23 parameters were negatively impacted. Okay, put simply, the bones are severely messed up. Terrible distortions to their architecture, terrible distortions to their bone mineral density. Anyone in the military before? No? Okay, because the person I work with on this used the term FUBAR. F-U-B-A-R, it's an acronym, and she said, they're FUBAR. And I didn't know what it meant, but apparently the polite version is intercourse beyond all recognition. So, how does this affect bone strength? Good news and bad. First of all, twisting force, the MOI, moment of inertia, as you can see, they're 60% weaker. So if you take a bone and you want to break it by twisting it, if you've got homocystinuria, it's going to break much easier. Frontal impact, again, 60% reduction in bone strength. Side impact, 60% decrease in bone strength. So I have a number of different lines of evidence indicating that this might be due to low folates. And so we treated both models with a combination of folate and taurine. We found the taurine made the Formate treatment much easier to bear for the mice over a long period. And what we found, the good news, is we were able to restore the bone strength. And basically, if you notice this bit here, the crucial bit here is that it is statistically no different from the control in the torsion strength, the frontal impact strength, and and where we go, and the side impact strength. So what is really interesting here, by the way, this is on a full methionine diet. What is really interesting here is that this works in the CBS null mice as well, where the homocysteine is not being lowered by the Formate. So as well as turbocharging the betaine response, it looks like the Formate is also improving the bone strength through a different mechanism. And the fact that it's not related to homocysteine explains why these bone problems persist even under good homocysteine control. So I do believe we're onto something. Okay, finally, I just wanted to mention why one of my colleagues, Curtis, in my department, had talked about a paper. He said, "Oh yeah, there's these two people. They've got the same mutation, but they've got a very different outcome." And we have to talk about polymorphisms and why people might respond to treatment differently, and two people with the same mutation may have different penetrance, might have different outcomes. And that is polymorphism. So this is for the non-scientist. When you've got a mutation, that's usually, it means it's a relatively rare thing. But a polymorphism is something that it's a variation, but the gene still works. And we all have polymorphisms. We all, some people handle caffeine better than others because of polymorphisms in the gene that metabolizes caffeine. Caffeine is an insect neurotoxin. We can only take it because we've got an enzyme that breaks it down. So some people can drink coffee and it has no effect. Some people can't sleep for days. That's down to polymorphisms. As an example, dihydrofolate reductase is highly polymorphic. Depending on which version of the gene you happen to be carrying, you can have a 7-fold difference in the level of dihydrofolate activity. That has the potential to strongly influence the kind of problems that you get, the kind of sequelae, like the bone problems, for instance, and also your response to treatment. If you've got one of the high levels of DHFR activity, you may respond to betaine far better than somebody with one of the really low ones. So, as well as DHFR, we've identified 10 different genes that are highly polymorphic with the potential to profoundly influence the pathogenic effects of homocystinuria and the response to treatment. More work in this area is needed, and it could lead to a kind of personalized medicine-based approach. Okay, so those are people that have worked in the various labs that I work with and my own. If you ever come to Denver, we have about an 80-foot statue there, which is what we're working for. Happy dancing homocystinurics. Okay, and for the patients, I just always want to stress that having a rare disease can be an isolating and bewildering thing. It can just be really, really confusing and awful. But I don't want you to let it knock you down. You're not alone. As you can see from this room and the room past the other side of the Berlin Wall over there, you're not alone, and we're all in this together. And we are making great progress with our science and translating this knowledge into improved understanding of the disease and designing better treatments. So hang in there, because good things are coming. And the reason I put my phone number and my email on there is if anybody you've got any questions about the disease or about our research, or you're confused about the treatment and you want to talk to me, I make this offer every time I give a talk. I'm happy to talk to anyone. Give me a little bit of time with my schedule and I'll arrange and I'll talk to you and I'll answer as many questions as I possibly can. Okay, thank you.